However, the model presents a puzzle. The genome is littered with thousands of GT and AG sequences, yet only a tiny fraction, less than 0.1%, were used as the actual splice size. This raises critical questions. How does a cell achieve such remarkable precision? This led us to our central research question: are there conserved signal sequences other than introns that help the spliceome recognize splice size quickly and accurately? To investigate this, we designed our model in 3 main phases. First, the model constructions. We built several deep learning models to predict the RNA splicing size. Second, model explanation. We used several technologies to look inside the model's black box and to understand why it made certain predictions. And third, biological significance. We took the patterns identified by our models and explored their evolutionary, functionality, and clinical relevance. Let's start with the model. First, we developed a deep learning model called Splice ULET. Its architecture is based on the ULET framework, which is good at image segmentation tasks. We treat the pre-messenger RNA sequence as a one-dimensional signal to be segmented into introns and exons. To better simulate the biological scanning process, we integrate bidirectional LSTM layers. The model takes a 1024 nucleotide sequence as input and outputs a probability score for each base in the central region, classifying it as a splice donor, acceptor, or lyser. How well does it perform? Splice ULET achieved an average intersection and union ratio of 0.84 on human transcript data. When compared direct with Splice AI, currently considered the SOTA model, Splice ULET showed superior accuracy under identical conditions. Furthermore, by using a sliding window approach, our model can accurately predict splice size in very long transcripts. In this page, we show that the PACE1 gene, which has 40 kilobases, our model can actually predict the splice size correctly. So we have an accurate model, but what has it learned? We employed a technique called GrandCam. This allows us to visualize which parts of the input sequence were most influential for the model's decisions. By looking at the model's internal layers, we can see where it paid attention when identified a splice site. The results were revealing. As you can see in this plot, the model's attention is very high in the intron region, immediately next to the splice size. So it is actually focused on the traditional intron signal GT and AG. And however, we noticed something else, something unexpected. There was a consistent and significant peak at the very end of the exon, immediately near to the splice donor site. This suggests that the terminal nucleotide of the exon were also critically important for the model's predictions. Interestingly, when we performed the same analysis of Splice AI, we found that it was also focused on the intron regions, but it did not show the distinct attention peak in the extron region. This suggests that our Splice ULET model might have captured a lower biological signal that other models had missed. To associate the role of this exon signal, we perform a new experiment. We create a modified model named splice unit CDS and train it exclusively on coding sequence, meaning that we remove all the introns. Just see the model can predict the join site of the mature messenger RNAs. So the model can predict well to find the joining sites. And when we visualize the model's decision process, we saw striking patterns. The model's attention progressively sharpened layer by layer until it focused almost on the final one or two nucleotides at the very end of the exon. To confirm this finding wasn't an artifact of one specific architecture. We employed two other distinct deep learning models. First, we used RNN, which was widely used in language processing, and we think the messenger RNA sequence can also be a language of the DNA or RNA. So we use the RNA to combine with a finite state automation to trace its decision paths. We found that over 80% of the decision paths that decision paths converge on a final terminal state only after reading the last base of the exon, which was overwhelmingly a G. Second, we use a transformer-based model, Robota, which is the architecture behind the large language models like BERT, ChatGPT. When we visualizing its attention mechanism layer by layer, we saw the exact same patterns. The models first focus on the GT, and its attention becomes increasingly focused on the single base at the very end of the exon. So to summarize the first part, we use three different deep learning architectures. When asked to find the splice sites, all independently coverage on the same conclusion. The terminal nucleotide of the exons play a critical and very important role in splice site recognition. This led us to our next questions. If the signal is so important, what is the sequence composition? We analyzed all human protein coding transcripts and quantified the nucleotide frequency at the end of the exons. The result was very striking. The single nucleotide right before the splice donor site is a G in over 80% of cases. Similarly, the first nucleotide of the next exon is a G, is nearly 50% of cases. So we can see that in the splicing sites, there is a motif that interesting is AGGT in the donor site and the AGGT in the accept site. This is very interesting. Then we look at the G usage in the context of gene complexity. We found that the genes with a relatively small number of exons, the usage of this terminal G is stable at around 80%. However, in genes with a very high number of exons, it is more than 70 splice sites, the usage of gene G increased to over 90%. This suggests that as genes become more complex, the reliance of the gene anchor point on this G anchor point for accurate the splicing is enhanced. This observation made us wonder, is this phenomenon conserved across evolution? We analyzed the gene terminal nucleotide usage in a very wide range of eukaryotes from simple yeast to humans. And indeed, we find that when it is in yeast, the G usage is around 30%, and in C. elegans, it rises to over 6%, and in vertebrates, it climbs to and stabilizes at around 80%. This progressive enrichment of terminal G suggests it confers a significant evolutionary advantage, likely by increased splicing fidelity as the organism continues to grow. To explore this evolution dynamic, we analyzed the homologous genes across species and characterized the species side as acquired, the splice side relative to the human transcript, definited splice sites relative to the human transcript, and conserved splice sites relative to the human transcript, and displaced splice sites related to the human transcript. And by clustering these position changes, we could stratify all human transcripts into 4 evolutionary categories, from basal to recent linkage. This clustering beautifully matches the knowing phylogenetic relationships, confirming that splice site positions have very actively remodeled throughout evolution. This analysis shows that the dynamic of splice site position are tightly synchronized with the evolutionary trajectory of species. Now let's connect this back to our terminal G. This heat map shows the evolution of the terminal G usage. Look down the diagonal. You can see that in species-specific genes, or orphan genes, the G usage rapidly increased from yeast to early vertebrates and then stabilized. Now, looking vertically up any column, you can see that for a set of homologous genes, the more evolutionarily advanced species always has a high G usage than the lower ones. This strongly indicates that persistent directional evolutionary pressure favors the usage of the terminal G. And we test this idea using our splice ULET models. We trained separate models on data from different species, and then test their ability to predict splice sites in other species. The results surely is very interesting. A model trained on an early species like a fly can predict its own splice size well, but failed to predict accuracy in a later species like a human. And a model trained on human data could not only predict human splice size accurately, but could also predict species size in earlier species with higher precision. We call that a downward compatible, upward incompatible paradigm. It mirrors the evolutionary trend of increasing complexity and reinforces the idea that the terminal G is a part of most splicing code that evolved later in evolution. So far, we focused on the DNA and RNA level, but this terminal G is within a coding sequence. So it means it must have influence at the protein level. A G at the end of exon constrains the possible codons at that position, which in turn influence the amino acid that can be encoded there. And it is exactly what we found. We have analyzed the amino acid frequency in splicing site. We saw a distinct preference for 5 amino acids: glycine, lysine, arginine. Just like the terminal G, the prevalence of this amino acid follows the same evolutionary trajectory, a rapid increase from invertebrates to vertebrates, followed by stabilization. This specific amino acid selected is determined by where the splice site falls with the codons. We classify that into 3 situations. One is C models, where the entire codon is preserved. The other one is F model, where the first nucleotide is preserved. And the S model, where the first 2 are preserved. For example, in F model, where the GGN codon is common, so glycine is highly enriched. In S model, AG codons favors arginine. This shows a beautiful co-evolution link between the splicing machinery need for G anchor and the resultant amino acid selections. This shows we wonder if this amino acid preference was driven by functional constraints like post-translational modification. However, we found no significant difference in PTM frequency at a splice site compared to a non-splice site. But we found the difference in the frequency of splice site in different functional domains. In domains with catalytic activities or architecture functions, the splicing site occurs more frequently. Protein kinase domain, peptide kinase domain, there is also has much splicing site. But in immune neuron development related domains, the splice site appears less frequently. In the catalytic function related domains, there are no dominant selections of the amino acids at the splicing model and the splice sites. In architecture function, like neuron development or net involving domains, there are obviously splicing patterns and amino acid dominant selections at splicing sites. We choose the GPCR as an example. The GPCR has 7 transmemory domains. We find there are differences in amino acids and splice patterns at different transmemory regions. First, in TM1 and TM2, which do not participate in ligand binding, and there is almost no splicing site. But in TM5, which need to bind the ligands through hydrophobic residues and abnormally use highly hydrophobic animal access at the splicing site. And the TM7 mainly binds to the polar group of the ligand, and its splice site use common hydrophilic residues. So let's take a summary of this part. The selection of splice site is not random, but is the result of the co-evolution of the spliceosome and the DNA sequences. In most cases, amino acids at splice sites have their own unique molecular languages related to splice site recognition selection, rather than just satisfy the degeneracy of amino acids themselves. This brings us to the final part of our story. We've established that the terminal G acts as an anchor for stable splicing. This leads to fantastic questions. Could the absence of G be a mechanism of generating diversity through alternative splicing, and could mutations at this position lead to disease? To answer this, we analyzed the transcript variants across six species. The results were clear and elegant. When two transcripts share the same start site but have different ending sites, the exon terminal G tends to use a variety basis like A, C, or T, but not G, to create this diversity. But in contrast, when exons have shared the same end site, or the common to all transcripts, G is widely used to ensure stability. So the terminal base acts as a switch, G for stability, and long G for variability. This has profound implications for disease. We analyzed SNVs for solid tumor sequence data. We found that when the terminal base is G, the probability of exon extension is very low. But if that G mutations to A, C, or T, the probability of the exon increase dramatically. The G anchor is lost, and the splicing machinery makes a mistake. So this is our finding. And here is a real-world example in the G-skip genes. Normally, splicing occurs at the end of the short exon, which terminates in a G. But in tumor samples, we find there is a switch from G to A, which results the downstream splice site is used instead of this position. So it results a long transcript. And our splice roulette models accurately predict this exact outcome. We found other examples of this mechanism creating novel disease-associated transcripts. So we think that the G is this clearly shows that the genetic effort is at the RNA level, not the protein levels. So to summarize this key section, we found that the use of the exon terminal base G guides the selection of different transcripts to some extent. In normal selections, the use of the base G at the end of the exon increases the stability of the exon, while the use of other bases produces more transcripts or proteins enriching diversity. In abnormal selections, if the original base G at the end of the exon is mutated to another base, the anchor will not lose its stabilizing effect on splicing. And regardless of the way the animal acid is changed, it may produce abnormal transcripts or proteins causing disease. To access this system, we analyzed over 9,000 exon terminal SNVs from the clinical VIR database. The patterns was unicorn. Mutations from G to other base were overwhelmingly classified as pathogenic. In contrast, mutation to a G were much more likely to be benign. We can find when similar mutations, the SNVs of ACT to G were all benign SNVs, but are among the SNVs of G to ACT. Nearly 70% were pathogenic SNVs. So it is very interesting. And we find that CleanVIR database has some experimental validated results that this SNV will result the splicing mistake. So we check the above SNVs with splice roulette. We found the exon change were consistent with the experimental results. So in fact, we then use splice roulette mode. We find that it was able to predict a whole spectrum of splicing abnormalities caused by these mutations, like exon skipping, intron retention, X extension, and whatever. Finally, we think that our model, splice roulette, can predict the SNVs caused splicing correctly. We constructed a disease network based on all the exon terminal G mutations. We observed interesting patterns. The disease was not randomly distributed, but increased clustered heavily in the cardiovascular and neurological systems. And in contrast, the endocrine system, digestive system, and other systems, abnormal splicing caused by exon terminal-based SNVs produce few and more dispersed disease. So this aligns perfectly with our earlier findings that genes with architecture functions, like those prevalent in the heart and brain, have highly conserved and inflexible splicing patterns. They are therefore much more sensitive to mutations that disrupt splicing. In contrast, genes in other systems with more complex interactions networks may have more tolerant for splicing mutants. So to conclude, our journey started with building a deep learning model to predict RNA splicing by using some explainable AI technologies. We uncovered a key role for the terminal nucleotide of the exon, which was previously overlooked. We then demonstrated that this feature, a preference of G in the end of exon, has been under strong position selection through evolution, likely to ensure splicing accuracy in increasing complex organisms. This gene echo has a profound biological significance. It co-evolved with the genetic code to influence amino acids preference at the splice sites, particularly in functional constraint protein domains. It acts as a switch to control alternative splicing G for stability and long-G for diversity. And critically, mutations that disrupt this terminal G as significant and previously underappreciated cause of human disease, especially in the cardiovascular and neurological systems. Our work integrates artificial intelligence with multi-level biological data to uncover new principles for splicing regulations. It expands our understanding from tissue-specific to functional-specific splicing and provides a new framework for interpreting the thousands of genetic variants of unknown significance. Looking forward, this suggests a deeper layer of information is encoded in our genome, a splicing code. We believe that by continuing to combine deep learning and biological, we can decipher more of this hidden language of life. Thanks my mentor, Professor Chen Yuzhang. He supports our project. Thanks Yan Shengkan, Zhou Xiaochun, and He Yixiao, which is contribute a lot to this project. And we thank Professor Zhang Zichao from Southeast University, which gave me much suggestions on splicing. And thanks for our collaborator in Institute of Automation, Professor He Ran, Cao Jie, gave me some suggestions on AI modeling. Thank you a lot. Thanks for your attention.
All right. Thanks. Great talk. Given the interest of time, we only have time for one quick, short question. Go ahead.
Thanks, great talk. I have two little questions. First one is I'm curious about the testing data. You say it's pre-MI and CDS. I'm curious about it's from reference genome, reference transcriptome.
Yeah, we use reference genome from ENCODE. Yeah.
Okay, thanks. My next question is the exome situation. Not about the business of splice, but extend the first exon or last exon, such as APA. It can produce a shorter UTR in some cancer cells, so some oncogene can be expressed faster. I wonder how can we find some rules in this situation?
I think the models, especially the accurate AI models, can predict the occurrence of the splicing mistakes. So maybe you can try our models later, and I think it may give you some help. Yeah.
Okay, thank you.
Okay.
Thanks. Let's thank our speaker again and move to the next part of the session, meet the editors, and let me welcome my colleagues on the stage.
So before the next talk, at 5 p.m., we will have a short panel discussion with all of the editors attending today. And I would like to thank all my fellow editors to give the best support for the conference and for this panel discussion. So this will be an open discussion with all of you guys. So if you have any questions, we will be happy to answer. So it would be great if you could first introduce yourself a little bit, let us know who you are and which area you are working on, and then you can tell us your question and we will be happy to answer. And it's also good to let us know who you want to ask. Thanks. Angela? Yeah, so first I will ask my colleague to introduce themselves.
Hello, I'm very honored to be here. This is a really fantastic meeting on a very emerging area of science. I'm Angela Eggleston. I'm a deputy editor at Nature. My background is biochemistry of DNA repair and transposition, so I'm definitely sort of a nucleic acid person. I have been in publishing now for 26 years. I started at Nature Cell Biology for 2 years. Then I was at Cell for 2 years, and now have been at Nature since 2003. So it's been quite a long career in publishing, but one that I've really, really enjoyed. It was always something I wanted to do, so I was very happy to get that initial job at NCB, having never taken a cell biology course in my life. So they put a lot of faith in me, and I'm very glad they did.
Hello, I'm Arunima Singh from Nature Methods. I've been at the journal for 6.5 years now. My background is in biochemistry and bioinformatics, and a lot of force field development, MD simulation, and looking at protein-protein interactions. And for the journal, I handle all of the protein science papers. So all the protein structure prediction, protein modeling, proteomics, and a lot of the glycobiology and lipidomics content. Yeah, and it's been a pleasure to be part of this conference as, helping to organize, select the speakers, and also attend. The talks have been wonderful, and looking forward to the discussion.
Hi, everyone. My name is Jay Pan. I am an editor at Nature Computational Science, kind of a new journal that launched in 2021. I was within the journal from the beginning, so I was within the company and the journal for more than 5 years. My background, I was trained as a physicist as my research background. I'm doing more molecular dynamics simulations for soft matter and also for inorganic systems. In the journal related to biology, I handle protein structure, structural biology related computational methods and AI related stuff as well. I'm looking forward to discuss more with you guys.
Hello, everyone. My name is Ophelia, and my Chinese name is Bu Yashan. I'm associate editor for communications biology, and which is also a new journal launched in 2017. And about my background is mainly about corneal inflammatory response and also its correlation with diabetes and gut microbiome, and also the use of bioengineered model to tackle this problem. So that's why I majorly handle bioengineering and biotechnology papers for our journal. And also it's my honor to be in Nanjing and in this conference. I'm looking forward to your questions as well. Thank you.
Good afternoon, everyone. I'm Sun Jiye, and I'm a senior editor of Nature Communications. My research background is I received my PhD working on the functional polymers, and followed by postdoc research on the preclinical evaluation of drugs and nanomedicine. At Nature Communications, I'm handling manuscript in the area related to biomaterials, bioengineering, and drug delivery. It's a great pleasure to be here, and I look forward to discussing with you. Thank you.
Good afternoon, everyone. Thanks for attending this amazing conference. My name is Wenbo Li. I'm an associate editor in Nature Communications in the structural biology, biochemistry, and biophysics team. By training, I'm a structural biologist. In specific, I'm an X-ray crystallographer, specializing for enzyme structure determination and time-resolved structure determinations. But then now I'm handling manuscripts in more and more areas, including in situ structure discovery by cryo-ET or cryo-EM, and also enzyme mechanisms, as well as any machine learning-based structure prediction and protein-protein interactions. I'm happy to take any questions related to this area and any other general questions around the editorial and publishing workflows. Thank you.
Thank you. Okay, now it's a question time. Maybe I will start with a question with Angela. So people would like to know, like, how do Nature editors see this new type of study, like AI biology papers, and what do you really expect from these papers?
So I think on the biology team, sort of the first papers that I can recall seeing in this field were handled by my colleague Tanguy Chouard. And so those were like the early papers from DeepMind, things like that. So not yet really sort of answering a biological question, but sort of setting the stage for what came later. And when Tanguy left the journal, I took over those areas again, which was basically protein folding and design. And the timing couldn't have been more perfect for me because he left just as the first AlphaFold paper was about to be accepted. So I accepted the first AlphaFold paper and then basically was there to handle AlphaFold 2 and 3. I have to say that at the time that we had AlphaFold 2 and I was handling that, we had no real AI editor at Nature. So I received a lot of help from the sort of fundamental particle physics editor, who at least could help me out a little bit with the background. And since publishing AlphaFold 2, like, my workload in terms of AI has just completely exploded because it was not only about sort of AlphaFold 2 in terms of protein structure prediction, but at the same time, on the protein design side, it was also expanding exponentially. So I was also handling all of David Baker's and all of the protein design field papers. So for me, you know, the past 5 years or so have been just like a very extremely exciting time period to be working in this area. Now we do actually have a dyed-in-the-wool AI editor on the physical sciences side. But in addition, sort of as you've seen from some of the more clinically oriented papers yesterday that were cited from Nature, it's really expanded into other areas, so clinical, genomics, and those are handled by other members of the team. So now it's, you know, not something that really is handled as, you know, AI, but it's handled as a way to answer a particular biological question. And so whichever editor sort of handles that area, they're the ones who have to sort of learn about the AI behind it. So I think it's a learning experience for us all.
Thank you, Angela. Any questions from the audience? Please get ready at the microphone, please.
Hi, everyone. My name is Jiang from Suzhou University. I would like first to send many thanks to organizer for providing platform to communicate with their editors face-to-face. My question is to any of their editors. It sounds much more difficult for me to convince their editors than to convince reviewers, because I got a lot of rejections directly from editor without any external reviewing. So I definitely know that the solid novelty of what I presented is the key. My question is there any other secret skills for us to convince editors to send friendly external reviewing? I mean, other than the novelty of the paper. Thank you.
Thank you. Who would like to answer?
I guess there's no easy answer to this, but I would say it's a combination of a variety of, like a combination of multiple factors. So it's the novelty of the approach, it's the problem that you are addressing, how you're solving it, the results that you get, how the data is analyzed. So it's a combination of everything, and each journal at the Nature family has a different bar, different criteria for gauging if the paper might be suitable for them and if we would like to send them out for review. A good idea, like if your concern is that your papers aren't being sent out for review as much as you would like, a good option might be pre-submission inquiries, because journals and editors do take the time to review your paper quickly to give feedback on whether the paper might be suitable for the journal or not. And then obviously when you meet editors, discussing your work and trying to figure out if the work might be suitable for the journal might be an option. But yeah, we do have, and with the new AI trend and the submission volume has gone up, but we do try to find time to reply to authors and queries of this nature.
Thank you, Dr. Singh.
You want to add something?
Yeah, I would like to add, because sometimes the feeling that if it's harder to convince editors compared to reviewers is that when a manuscript is already with reviewers, that means it has already passed assessment or it has already been sent to reviewers by the editors. So that means the first sort of filter is already done. So maybe that's a sort of illusion from the feeling that why it's always harder to convince editors. And I feel the trick, maybe it's not a secret trick, it's maybe a very public trick, that how to make editors be convinced about the importance of studies, that to try to make where the novelty is for a study very clear in the paper, and also how the data is valid, and also why it is important to the readership of the journal, depending on which journal you are submitting. I think that's the important thing to sort of convince editors in a very initial stage. Thanks.
Thank you, Sophia.
Thanks for the question. And then further to, I'm in the life and physical sciences team of the interfaces team of nature communications, and primarily I'm handling manuscript at the interfaces of different disciplines. For interdisciplinary manuscript, we not only look at the conceptual novelties. Instead, we perform an overall assessment of one manuscript, and then, as my colleague has mentioned, we consider multi-factors, including the relevance to the journal, compliance with the journal's editorial policies. For example, related to clinical trials, we have very strict policies. And also the strength of the evidence supporting the major claims, as well as the advances compared to the state of art. And specifically, and for the biology-related started, we look at the conceptual novelty and the mechanisms insights. Importantly, we look at the biological and physiological validation, as well as the preclinical, clinical relevance, as well as potential benefits.
Thank you, Dr.
Thank you. Next, please.
Okay, yeah, first of all, I'm just, it's my great honor to be here to have a chance to talk with the editor. And I just kind of enjoy like submission, the whole submission process in the Nature journal. So even though sometimes you will meet some tough like reviewer, but your assistant really make our life easier. So I really appreciate that. So my quick question is like, so now I have been working on AI for like multi-omics modeling for a long time, but inspired by the ChatGPT, nowadays I think there's a lot of hype in the AI for biology. People always like say that they're able to solve the virtual cell using some foundation model. So I really want to like have your comments that what kind of a model that paper can be sent to the peer review from your perspective. I think that's you are kind of like people controlling this field, that what kind of people are really using the AI to solve the problem. So I really wish to have your comments. Yeah, thanks.
Jie, do you want to comment?
So for us, Nature Computational Science, we are publishing a lot of computational tools. So AI is one part of computational methods. So the question we always ask, no matter what the specific research area or domain it is, what unique capability you provide based on your development of the tool. Right? If you could calculate something more accurate, or you can calculate something more efficient, that's the thing we will look at. And even and the accuracy improvement or efficiency improvement that need to be landed at some practical problems, like why this is matters to important emerging topics in your domain, right? So the practical application or the demonstration in practical problems, that's something we will also look into whether that is a timely and important topic. So to answer that question past the editorial first assessment, I will see if you claim accuracy improvement, provide us with the benchmark. If you say it's scale better, right? The method scales better in terms of size or it's efficient, then also provide us with the benchmark and also tell us why this improvement is important to the field. So that's what my answer to the question.
Okay, thank you.
Yeah, I would like to add something, like I echo on what Jie has mentioned, because at Nature Communications, we also receive a lot of computational biology papers. But because we are a multidisciplinary journal and we would like to have a broad readership, sometimes we don't just want to see like a fancy model, like how you describe your model with different hidden layers, or you just describe your model as complex as possible. But we also wanted to see like what exactly is the biological question you're addressing. For example, if you're just repeating somebody else's work, for example, you just do a spatial deconvolution of the transcriptomics data. A lot of people are doing this. So from our eyes, we feel like this is not a really timely or novel question, but this is something, it is challenging. There's no doubt it is a challenging, and it's still some new methods are needed. But we really want to see from which angle or why you think that your method is addressing the question in a proper or like a better way. Another thing, as Jie mentioned, is we also care a lot about applications, especially for nature communications. We wanted to see how your model can be applied to a variety of different data sets or different cases. So we hope you can demonstrate your model in different case studies using a variety of real-world data sets and also like synthetic data sets. Angela, do you have something to comment? Any more comments? No? Yeah. Thank you.
Thank you. Sorry to forgot to introduce myself. Like, I'm Jie Cong from the Chomping Lab and wish to cooperate with all the editors in the future. Thanks.
Thank you.
Hello, this is Kai speaking. So I'm coming from Xi'an Jiaotong University, and I gave a talk about sequence to image in the morning and the last speaker in the morning session. And so I have two questions. The first one is that in nature journals, there are actually a lot of different, like nature method, and nature technology. I think a lot of the journals actually fit the topic like computational AI. And sometimes we found it very difficult to find which journals to submit to. And is it possible that we can have some kind of the guidance, maybe an AI portal that can automatically get an assessment which journal should we submit first? So this is the first question. So the second question is that, and what's in terms of I think during the review process, so reviewers are really powerful, and in a way that sometimes can dictate the future of the paper. And once that I had one paper that we are using we are using computer science technology, data mining, and one of the reviewers saying that he prefers statistical method, and there's no way we can meet in the middle because we have to turn around, rewrite everything, and simply because of this, and paper was rejected. I think at some point the editor may step in. And so some comments are doable, sometimes not doable, I think. And so I just hope that the remove process can be smoother. I think I had a very wonderful experience with nature journals. Thank you.
Thank you. So for the first question, probably it is for Jie and Arunima. It can be something like, how does your journal differ from Nature Biotech regarding the computational biology papers? And how does your journal differ from Nature Machine Intelligence regarding computational biology papers?
All right, let me start. This is not an easy question. So for us, I think, like in one of the last talk of quantum computing with biology problems, the question we will ask is what computational insight you provide, right? That's the key or the most major finding our eyes will look for. Of course, journals has overlaps. Like we also publish applied work, but our mainstream, for example, computational science, we are mainly looking for papers that provide the strike to drive the computational science. Of course, like I said, application is important to kind of demonstrate why your computational or methodological development is important to timely topics or domains. But that's the question we'll ask. But maybe, for example, biotech may be more focused on those maybe translational applications. That I cannot speak on behalf of Nature Biotech, but that's maybe the slight difference that we are looking at. Also, in terms of Nature Computational Science and another journal, Nature and Machine Intelligence, we have a lot of overlaps. We both publish machine learning, AI-related stuff, but the clear distinction is like we don't publish robotics, like those robotic engineering or more hardware-based papers, but they do publish the robotics. So that's kind of we have overlaps, we publish similar topics, but we have some distinctions. That would be my answer. Hope that's helpful.
Yeah, so to add to that, between nature methods and nature biotech, again, there is a pretty heavy overlap, but as Jia mentioned, nature biotech also considers translational applications, while nature methods is focused on methods for basic biological research. So if your method can do both, it is up to you to decide where you want to submit. So if you want to pitch the story for more translational side, then nature biotech might be a better option. If basic biological research, basic biological understanding is the stronger aspect of your method, then you can submit to us. But I see a lot of papers that Nature Biotech has published that if we had gotten the paper, we would have sent it out for review. And I see a lot of papers that we have published that could have gotten published at Nature Biotech. It depends on what the strongest aspect of the paper is. We get a lot of papers that have very innovative computational method development, but they don't have very strong biological applications. And so for us, the actual biological finding is the more important aspect. But if the computational side is innovative and exciting, we frequently consult with other journals that might have a different bar. So like Nature Computational Science or Nature Machine Intelligence might want to focus, might focus more on the method development instead of focusing on what the biological outcomes might be. So we have internal consultations for papers like that. And then again, you also have to consider your target audience, who is reading what papers. So for nature methods, a lot of biologists are reading our papers, so we tend to focus on that. But a more computational focused journal will obviously have a slightly different audience.
Thanks. Any other comments? Ophelia?
I just have some very general input for the second question. I think it's not only one case. It's maybe more than one author may have experienced this. So first, a general input is that it's always the editor making a decision based on review reports. So we will check the review reports, but the decision is always coming from the editors. So that means when there is a concern about the review reports, for instance, there are really technical or advice on the reports that is not actually feasible or in the scope of the study, it's always welcome that you discuss this with the editor via email, because although we make the decision based on review reports, but sometimes we cannot imagine maybe every single difficulty the author may face during the revision. So maybe next time in such a situation, it's always helpful that to raise this concern during revision, so that we may take this as a reference when consider the reviewer comments and also making a decision. Yeah, not sure if there's an input.
So I would say for regarding a bit a bit. So for those questions, comments, if you think you can suitably address them, and then you could do it through the experience, more experimental or more evidence. And if you feel you are unable to address it, be transparent about it and write in the responses to the point responses to the reviews. If you are uncertain, and then you can always reach out to the editor, and then the editor will guide you through the revision process.
Okay, thank you.
Thank you. Next one.
Good afternoon, all the editors, and my name is Xing Guo. I'm from the Shanghai Academy of Artificial Intelligence for Science. So we are a very new institute, and we established 2 years ago. So my major research is focused on the AI and how to apply it to the multi-omics analysis. So I actually have 2 general questions I would like to ask. The first, I think the Nature series is more focused on the new trend or something new, the new technologies. And since we are in a very fast changing world, and how do you guys to keep up with the latest knowledge with your professional background, and how do you choose to learn something new that helps you to determine which topics is suitable for the publication? And the second question is about, definitely we have some topics that is not suitable for the publication, and definitely we will have something is suitable. So how do you choose over one topic over others? And could you maybe share some examples in your editorial history? Thank you.
Yeah, thank you. For the first question, probably Angela can answer. As we Chinese say, it's never too old to learn, right? So it's always like a lifelong learning.
Yeah, I think in a way it's sort of driven by what comes across your desk. So as Aranima said, you know, we are pretty busy all the time. And so, you know, what I receive as submissions, basically, in me having to do the background to understand the paper, sort of keeps me on top. And obviously, people are submitting their best work to us. So, you know, I am seeing the best work sort of as it's being submitted to me. Obviously, it gets published by other journals as well, but that will come up in the background I do when I'm looking at the manuscripts that I receive. It is kind of difficult because there are so many papers that are sort of doing similar things but getting slightly better benchmarks. And it's like, at what point do you say, you know, this is now something I'm going to send out to review or not? In a way, to me, it sort of harkens back to when cryo-EM was first being done, and the first structures were ribosomes because they were big and easy to look at. And, you know, I would see papers from the same groups, you know, 2 months apart where, you know, it was now, you know, 0.4 angstrom better resolution. And, you know, because the field was moving so fast at that time, and you could actually get new information from that 0.4 angstroms, you know, we would send them out to review. And you know, as reviewing them, reviewers would say, well, but I'm sure they're going to have something better, you know, a few months down the road. It's like, well, but I can only, you know, really assess what is here in front of me now, and not, you know, hold and wait for something better to come along. So, you have to sort of make the decision based upon sort of your past experience with other papers because you want to be consistent. But for me, what I'm really looking for is something that sort of the authors have been able to convey to me sort of the significance of the advance. So, this really is going to have some kind of impact. It's answering an unmet need, and it will be able to do something that is not currently available. So that's sort of how I make the judgment as to whether or not to send it out to review.
And as far as keeping up with the field, as Angela said, we are reading a lot of relevant research in the space that has been recently published, and then attending conferences. That's one of the reasons we come to conferences, is to get an idea of what people are excited about, what are the pain points in a field that everyone is trying to address, and where exactly is the next innovation waiting to happen. So just attending conferences and trying to identify those points.
For the second question, how do you compare two studies? Do you want to comment on anything?
I just want to comment on the first one first. So I think sometimes I keep up with the field by reading constructive referee reports. That's very helpful sometimes to understand the field better. So please write us constructive review reports. That's better for our editors also to make a most informed decision, and also helpful to the authors, even it is a negative results.
Thank you.
And what's your second question? Sorry, I passed.
So how do you choose maybe one study over another one? What's the, for example, the topic it's about, or the writing, or the illustration, etc.? Maybe you can share.
You mean the topic, or you mean the?
I think I would more interested in the topic. Yeah.
So we don't have a clear bias on the topic because, as Angela and Angela just said, we just work on whatever that submission comes in. If we feel this is an important field and it's a timely topic, we will consider it. That's would be my simple, maybe not a helpful answer.
Thank you. Anyway.
Anyone want to comment?
Especially in new upcoming fields, I think we also tend to invite papers. So for example, more recently, I don't handle this area, but another colleague, she handles lineage tracing. And in the last few years, we have seen some very strong papers, and Nature Methods, for example, had not published much in this space. So obviously, reaching out to the community, doing lab visits or attending conferences and inviting papers is another way of how we expand our scope at different journals. And as far as choosing a topic, again, depends on how exciting, how new, how innovative something is. We have, at Nature Methods, we want to see a substantial performance advance as well as conceptual advance for new methods. So, like Angela said, in cryo-EM, when you saw, when the field was new, you saw you got a lot of new information from very incremental resolution improvement. But as that got saturated, we wanted to see more, substantial improvement in the results. And we have published a lot of papers in that space where the sample preparation has become easier, the resolutions have improved substantially. Another example would be in proteomics. For single-cell proteomics, we were seeing, at least 3 years ago, we were seeing 2,000 to 3,000 peptides identified with very high confidence. And now with the development of the Astral mass spectrometer, we are seeing closer to 5,000. So we also look for jumps in the field of that nature.
Thank you.
Thank you.
So this also reminds me of another question that always comes to me, do you have a preference of specific field? Because sometimes we do publish a lot in some field, but doesn't mean we have a bias on it. It most likely because we receive a lot of submissions in that domain, And if you really count the acceptance rate, that might be lower than other fields. So that's kind of what you observe, but it's not exactly we are biased, but just like some of the fields are evolving very fast, and they have a lot of papers produced during the short time period. So kind of answer what kind of a question, yeah.
Thank you.
Thank you. Next one.
Good afternoon, everyone. My name is Bin Zhang, and currently I'm an assistant professor of computational biology at Mohammed bin Zayed University of Artificial Intelligence. So it's really great to have these opportunities to meet so many editors. And the questions I would like to ask, or I would like to hear your opinions, is one of my real case. So actually, I have submitted several manuscripts to Nature, and unfortunately, and all of them got a desk rejection. But one time, and in the email, the editor mentioned like it's a single cell study, and they mentioned that the sample size is too small. So I don't know like whether this is like have some indications the editor is like also have some interest about our study or not. And I would like to hear your opinions about this scenario. Thank you.
Well, if it was single-cell transcriptomics, that was not me. I don't know where, the editor who would handle that sets the bar. He or she will know sort of what they're looking for. Generally, if they give you some kind of feedback like that, that's pretty specific, like, the sample size being too small, I would say probably they had some interest in it, and just, in terms of sort of the methodological details, it wasn't quite there yet. So I don't know for sure, but that would be my guess.
Thank you.
Hi, Valentine Svensson from Tahoe Therapeutics in San Francisco. You mentioned spatial deconvolution as something that a lot of people are submitting, but the increments are small. I was wondering if there's some other examples of problems that people are trying to solve with AI that you're getting a lot of submissions for, but they're all incremental improvements, and you're a bit tired of these incremental changes, and where you would like to see larger step changes?
Because I handle like half of my papers are mostly about single-cell genomics, spatialomics, and also some genetics papers. So these questions are quite commonly addressed by authors. In other cases, I think I have seen some machine learning-based platform papers. They would like to use machine learning models to analyze experiment data, or they want to optimize some experimental protocols. We sometimes see like people want to emphasize the component of machine learning in the work, but we would like to ask the author: is it really need? Is this something that we must have for a machine learning component, or it is already well established? I think that is something that I always see. Like to me, it looks like quite incremental. But I also want to hear others. Have you seen any other papers that many people use AI to address?
Thanks for your question. So with your question, the incremental advances, we did have the discussion with our internal teams. One of the point we were thinking is between submission to publishing, the general timeline is about around 6 months time. So then now the machine learning AI paper are a very fast evolving field. By the time you submit the paper to the time the paper has been published, I mean, if it's incremental advances, then the bioarchive, although, as a Nature Communications, we do not consider bioarchive as novelty, but then the advance will be dramatically decreased. So, but then, advance can be think in different ways. We are not only see just one single part. We're seeing the bigger picture. For example, if the algorithm performance is increased slightly, but then your application might have a broader range of application, then we may also consider it.
But are there like particular problems that everyone is submitting to solve, but they're kind of solved already?
I think different journal has different policies. Definitely with Nature, I mean, I can't say that, but then with Nature Communications, we do have a anti-scoop policy that we are really proud of. I mean, we really want to to become a host of all these kind of great works, and then to publish it. So our goal is to publish your work, and then, I mean, as I always say, we do filter, enhance, and amplify. So, so for example, if some similar work has been published on bioRxiv, as I mentioned, we won't consider that as diminish of your novelty. So I hope that answers your question.
Yeah, sure. Was hoping for just like a specific like topic that like I'm really tired of spatial deconvolution or something.
Thanks.
Next one.
Good afternoon, editors. First, thank you for organizing this great discussion. I'm Zais Zhang from Princeton University. My research focuses on AI for biosciences, and recently have been working on the safety and the security issues of AI models for biology. I have a problem. So recently, for example, Science published a paper showing that AI protein design models could bypass safety screening. So I would like to ask, how does the Nature family journals view this emerging area of AI model biosafety and biosecurity? Do you see it becoming an editorial priority or one emerging direction for future publications? Thank you.
This is a relatively, and actually a very new area, so we haven't gotten any papers in this space, but I think it is an important area. So like with any new space, I think we'll have to probably have an internal discussion within the Nature journals about the effect these might have. We don't have a policy yet, as far as I know. But we'll probably have a discussion about it in due course of time.
Yeah, I think it is attracting our attention as well. We are looking at these research contents that we call it like a double use research content. It can be good, or sometimes you can use it in a bad way. We recently published a paper called "The Risk of AI Scientists," and we feel like if we ask the large language model to answer something, if we can do some like a jailbreak, we give a prompt saying, please give us the step to generate a toxic compound. Probably it will be blocked. But if you ask a specific toxic compound, it will probably give you the answer. This is something that we are looking at. We want to, probably in the next following months, we will have our policy for these type of papers. Yeah. Thank you.
Thank you.
Next one.
Okay. Good afternoon. I'm Xiao Mingyin, a speaker from morning session. I have submitted several manuscripts to Nature journals, and all the editors are quite nice. Thank you so much. My first question is, I feel that the competition in every field is quite intense nowadays. And what kind of manuscript would you prefer to peer review?
Maybe I can give a general answer first. So first, in a very general sense, we prefer works that provide novel insights and also with valid data to be sent to peer reviews. And as you can tell, since we're from different journals, so that means the different journals may have different bars for their understanding of novelty. So that is a question, what type of journal you are aiming for your study. Maybe some of my colleagues can give more concrete answer in a more specific sense.
Yeah, I guess the bar is different at every journal. So for us, it's novelty, impact, performance, how the benchmarking looks across other comparable tools, and if it provides biological insights that were not available through other methods. So we essentially look for that.
Okay, my second question is, I imagine that editors at Nature journals receive a high volume of manuscripts every day. Could you tell us which research areas are currently saturated with very similar submissions?
Because of time, I will give a very brief answer. I receive a lot of papers on single-cell data clustering, and they still develop different clustering methods for single-cell RNA-seq data sets, and they claim that their method is better than the state-of-the-art. But we are not really convinced by these papers because we feel like this field is already saturated, and the performance doesn't really show like a substantial improvement compared to the SOTA method. And unfortunately, we have to reject these papers. But because we are running out of time for this session, and we know that you have many great questions for us, and we want to thank you for your support, either as authors or as reviewers, because our journals are highly supported by all of you, and we can run in to be the best portfolio ever. And we really hope in the future we can publish many more of your work in our Nature portfolio journals. And I want to thank my colleagues for the great answers for this panel discussion. And now let's continue with our speakers. Thank you, everyone.
All right, let's get back to the scientific sessions. The next talk is going to be a virtual talk from Nicholas Cather. The title of his talk is AI in Precision Oncology and Cancer Research. Let me know when Jacob is ready.
Hello.
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 Hello, the floor is yours.
Thank you very much. Thank you very much for the kind invitation. My apologies that I cannot be there in person, just remotely here. But I see all of you, I see you on the camera, and thank you for your interest. So my background is, I'm a medical doctor, I'm an oncologist, but also I'm a professor in computer science. And I will speak about our research that is AI in precision oncology and cancer research. So here are my disclosures. So the field of artificial intelligence and cancer research has evolved over the last decade, has evolved tremendously. When I started in this field, we were working with what we now call classical machine learning methods, where you take data sets with specific predefined features, and then you can train classifiers or predictive models on top. These methods are still valuable, but they are very limited in a way that many of the real-world problems that we face cannot be addressed by them. So in the 2010s, specifically in 2012, the field of medical data analysis got a huge boost through the availability of deep learning with deep convolutional neural networks, specifically in image processing, which allowed us to solve many of the questions that we could not solve before because we could not basically predefine the features that the classifiers should look at. Now, recently in the 2020s, based on the successes in self-supervised learning, we can now train models on unlabeled data or poorly labeled data with a lot of noise even. And then we get a foundation model that we can then use for downstream tasks. And this is basically for us in many steps, in many projects now, the gold standard and the standard method. Another type of foundation model is large language models that were trained on the whole text that's available in the internet. And so they can be reused repurposed for many, many tasks. Also, because they were trained on language, they can be embedded in chatbot interfaces so that you can immediately talk to them without having another model on top. So this is very powerful, and it becomes even more powerful when you couple it to other software components. Then you get what we call AI agents, where you have a large language model that can control other softwares and other models and can perform stepwise reasoning and can therefore solve complex tasks. I will give you some examples for along this spectrum. First, let me start with a topic that we are working in for many years that we call AI-based biomarkers. Specifically, we are interested in pathology images of cancer, which are available for every single patient with cancer. And one of the first works in this field that we did back in 2019 was that we hypothesized that pathology slides of cancer can be used for computational image analysis, and not only to make the diagnosis of cancer, but also to predict genetic alterations of the cancer. And so here we hypothesized that for colorectal cancer, microsatellite instability could be detected with deep learning. And indeed, this worked very well. So we were able to train deep learning models to predict microsatellite instability. And this is a very important biomarker, because if your cancer has this, then you can be treated with immunotherapy. So this is now approved as medical devices by several companies. So it has really stood the test of time. On the other hand, you can use the same approach for prognostic biomarkers. You can train deep learning models on pathology slides to predict patient survival and outcomes. And we have done this in many studies, and many other studies have also done it. What is very interesting is to take these predictions and then combine them with other biomarkers. For example, with ctDNA in colorectal cancer. So in this recent study, we took pathology slides of cancer. We trained a deep learning model on these slides to predict relapse risk and survival. And then we combined this with a ctDNA, circulating tumor DNA from the bloodstream, to a combined biomarker. And this biomarker really had an excellent performance. As you can see here, it's a near perfect prediction of outcomes in these patients. There's many other biomarkers that you can also predict with this approach from pathology slides, and one of them is homologous repair recombination deficiency. That's another biomarker that occurs across multiple cancer types that we can detect with deep learning based on these images alone. And finally, we have also, together with colleagues from the United States, from Memorial Sloan, we have recently published this, where we took in breast cancer deep learning-based image analysis of the tumor tissue, combined it with language analysis to one single model, and then predict the risk of relapse. And this worked basically very well and better than basically genetic methods of RNA expression signatures. So we have recently in our professional society, the European Society for Medical Oncology, we have convened a guideline committee, and we are working on guidelines for how to implement these biomarkers in clinical care in cancer. We propose 3 classes. Class A is AI for biomarker quantification. Class B is prediction of existing biomarkers from other data, for example, predicting microsatellite instability from pathology slides. And Class C is novel biomarkers, where we predict outcomes and treatment response directly. So this will come out hopefully soon in Annals of Oncology, the ESMO journal in this case. Let me switch to the other part. So I hope I could show you that AI-based biomarkers can really be a very interesting approach for precision medicine, and you can use deep learning not just to make a diagnosis of a cancer, but also to extract more properties, molecular properties, or clinical outcomes. Now, large language models are the other side of the spectrum, right? This is basically what most of the technological progress in recent time was about. And in my group, we started working with large language models back in 2022, 1 year approximately before ChatGPT came. At that time, there was a model called GPT-3 that was basically just an autocomplete model. So you had this text box on the internet where we typed text, typed a question, and then you could click autocomplete, and then the model would create this green text. And so I think that our attempts here were probably the first attempts to test the capabilities of such a natural language processing model for precision oncology knowledge in this generation of these GPT-3 generation models. By the way, back then we submitted this. We tried to submit this to Nature Medicine, but it didn't work out. So basically, sometimes you're lucky, sometimes you're not lucky, right? But of course, this idea of large language models in oncology has evolved tremendously, where we now have, everybody has large language models on their phone, right? We're all using them every day. There's different ways how to use them. For example, in oncology, one is zero shot. You just ask a question and you get a response by the model, which is based on the model learnings that it has from its training data, right? However, this is a bit limited because it's not up to date, and there's also many other limitations which are solved by what is now becoming the standard, that these large language models get the context from websites, right? So you have additional context here. But the problem is that these can be like everything. It can be scientific articles, but it can also be like press releases and other non-scientific sources. So a very safe way to approach this is to provide explicit context. So you take, for example, a guideline, medical guideline, you upload it to ChatGPT or a large language model, and then you ask a question based on this guideline. And as we have shown in this study here, this works really well for oncology decision making. So, of course, large language models are now being used by everybody, right? Even in oncology, and there is very interesting things are happening. For example, when the new GPT-5 was released a few weeks ago, then they released this basically, and they took a breast cancer patient on the stage and had an interview with her about her breast cancer and how a large language model helps her to understand her disease. So the application of this technology for cancer is really on the making headlines, right? And there's also increasingly good evidence coming out that these models are becoming really good. I think that one of the most interesting studies was here in the top middle, where the authors report a randomized prospective trial of using large language models as psychotherapy support for mental health treatment, and this study met its primary endpoint. So increasingly, we also have clinical evidence for what we are doing. So again, in our professional society, ESMO, we have published, and this was published a few days ago in Annals of Oncology, where we showed the first guideline for the use of large language models in oncology clinical practice, which is called LCAP, where we provide guidance how to use this this technology for patient-facing apps, physician-facing apps, and background applications. But I think the most interesting part about large language models is that they enable us to have agents, right? So you have a large language model that can control other software and can interact with it, and we call these other software types tools. And then, if you have a large language model that has access to other tools, what you have is an agent that can fulfill a high-level request by requesting the help of specific tools. So this is, of course, impacting many areas of our society. For example, software development, where agents are becoming the norm for many things. For example, in many things like user interface, etc., the software development agents can do a very good job, right? And on the other side, of course, also for biomedical data science. So we published this here last year, this concept of how autonomous research agents could help us. Of course, we've heard in the preceding sessions, in the preceding discussion, we've heard a lot about biomedical research agents. Now this topic is really coming into the mainstream. I think here this was early 25 when we conceptualized this here in Nature Cancer. And I think that many of the tasks that we are doing nowadays in biomedical data science can be potentially agentized. One thing that can be agentized is clinical decision making in oncology, specifically in tumor boards. So here, a short while ago, this summer, we published for the first time a tumor board agent in Nature Cancer, where we have cases from a tumor board where you have clinical history, radiology scans at multiple time points, you have pathology images, you have genomic data, and then this is very difficult to put it all together into one single machine learning model. But if you have a large language model that can look at the case and then can call specific models that analyze specific types of data, you get a very good performance. And this is then, and this can then even automate a whole tumor board, as we have shown, and compared to a set of human doctors here in this study. There's another study that is currently still under review at one of these Nature journals, where we take this agent and we give it autonomous access to an electronic health record to basically actually perform complex tasks that human doctors, sequential of tasks that human doctors would do. So this is basically work in progress and hopefully to be shared very soon. With that, I'm already at the end of my 15-minute presentation here. I hope we have maybe some opportunity for questions. If I may take this point here to point you to another potentially interesting conference in the ESMO AI conference in Berlin in a couple of weeks, where we'll also discuss these topics. And with that, I want to close and thank you for your attention and thank my research team. We are an interdisciplinary research team based in Germany. About half of the team are medical doctors or biologists. The other half are computer scientists or engineers. We have some of my best students and postdocs are from China. And of course, this is a completely international team. So if you are, we speak English all the time, at all times, right? So if you are interested in working with us, please reach out. And with that, I want to thank you for your attention, and maybe we can have a few questions if this is appropriate. Thank you.
All right, thank you, Jacob. Can you hear me well? Here as well?
Yes, I can hear you well. Yeah.
Any questions from the audience? Go ahead.
Hello, wonderful talk. And I have a brief question. You mentioned the agentize the AI. I just wondering, is it possible in the future the like AI agent can take responsibilities, or does at the end of the day does a human take the responsibility?
Yes, this is an excellent question. Right now, the situation is clear, at least in Europe and in the US, that a human should take responsibility. And this is how all the legal, basically the laws and regulation is set up, that agents are AI software, so they count as a medical device. In the US, they would require FDA approval, and in Europe, they would require a CE mark. And basically, a human would usually take responsibility. I think that as these agents are becoming more and more autonomous, there might be some cases that don't have to be seen by a human at all, right? So then we have to come up with new rules. Who can have the responsibility in this? In this case, right now, it's very clear. It's like, yeah. But I think this is a topic that we need to debate for the future.
Thank you.
Thank you.
All right. Any other questions? No? Then let's thank our speaker. Thank you, Jacob.
Thank you very much. Have a good conference.
We are a bit early on the schedule. The next two talks will be flash talks. The first one is from Huang Shuo, Nanjing University. The title is AI-assisted nanopore multiomics. All right, let's welcome our speaker.
All right, thanks everyone. My name is Shuo Huang. I'm a junior faculty member from Nanjing University. So I'm specialized with nanopore research. So today I'm going to tell you something about nanopore and how AI would help nanopore to move further. So if you happen to know nanopore, well, it's like that. It's very simple. If a molecule can translocate through the pore, it will produce like signals like that. By analyzing the open and closing states, you will know what kind of molecule was just passing through the pore. So the concept is very simple, but actually the most striking application of nanopore is nanopore sequencing of DNA and RNA. So it's quite different from well, conventional nanopore sensing, because like Oxford nanopore technologies have already commercialized the device. And from that, we will see data like that. It's very complicated looking, and AI expert will be really excited to analyze this part of data. So, but actually, even for Oxford nanopore technology, there are still many remaining challenges that either their device or their technologies can't do, and then these become our research. For example, like metabolomics and glycomics and proteomics. So according to these figures, you can see that they are very complicated, containing like a rich chemical information, just like a biology language. So we will need like a much improved resolution of nanopore and also the artificial intelligence to solve these problems. So by looking at more conventional and old-fashioned nanopore research, on the left side, you will see like these are nanopore research data, right? So they look very simple, and for AI experts, we will not get excited by looking at this data because it's just too simple and doesn't contain too much information. So what I aim to do is to engineer the nanopore so that it will contain more information so that AI will become more useful and my data will be more suitable for AI. So my data will look like that. Just for like one single compound translocation, and it will just look like that, and the data contains a lot of information in these noise patterns. So we will need like more parameters to describe one single event so that it can simultaneously analyze multiple or large variety of analyses without interfering from each other. So that's the key. So before I talk about nanopore glycomics, I would like to tell everyone that's a truth that most people actually don't know. A nanopore, besides can be used as a sequencer, it could also be used as a single molecule reactor. For example, you can monitor the happening of this single molecule reaction just by looking at the data below. That contains a lot of information and very high resolution. Even by the addition of one atom or the deletion of one atom, we will tell the difference. So basically, it contains rich data features. And with that kind of resolution, we think we are ready to go to a more challenging problem. For example, glycomics. So for these monosaccharides, they look almost identical to each other, and their molecular weights are pretty much the same. So they are very difficult to be discriminated. So what we want to do is we have to specially engineer the pore so that one of the monomer is different from the other, so that you can guarantee the placement of a single chemical reactivity site. For example, in this time, there's a phenylborotic acid will be put to the pore constriction, and then a highly reversible reaction between the sugar and the phenylborotic acid will happen, and this could be used to much improve your resolution for saccharide sensing. So when we did that, we did get this heterooctameric MSP nanopore done, and it's working really good for discrimination of different monosaccharides. At that moment, I was really excited. So I think this is going to be a really huge paper. So I submitted to all the high-impact journals that I know. But actually, I got 14 rejections in a row. And then was peer-reviewed by PNAS, and my review comment said, I don't believe AI at all. So at that time, people are not really confident with AI. But eventually, it was published by Angewandte Chemie, and here I would just use like one animation to show you how it is working. So technically, this is the heterooctameric MSP pore that I mentioned. The reason I want to make it like one of the monomer different from the other is to guarantee the placement of a single like reactive site, which is a phenylborotic acid. And after modification, the pore could be used just like a regular pore. You don't need to do any extra things. It will report events like for L-sorbose. Remember this feature. And then this is the D-fructose. So they are different, right? So AI will be used to recognize them. And then this is the galactose. And because the pore has a really high resolution, so you can just put all kinds of monosaccharides into this same assay, and they will tell you the result. So eventually, we will have like a secondary question. So the glycan could be complicated because different, like monosaccharides could be linked in a different way. So that will generate a lot of isomers. But actually for this pool, which is very sensitive to this kind of isomerization, so we can actually give discrimination of all of them. And when this paper was published, Nature Chemical Biology has written a research highlight by describing this technique, nanopore-glycan profiling. So we were really proud to be the first of its kind and could help nanopore, the field, to get more attention on nanopore-glycan sequencing or nanopore-glycan analysis. So after that, because we already engineered a technique that is sensitive to saccharides, actually you know that for RNA nucleoside monophosphate, it also contains a saccharide subunit, which is the ribose part. So the ribose will bind with this pore and will generate like C, U, A, and G. So it's 4 distinct signals, which corresponds to the RNA base. If we have this kind of high resolution, then why not just go to like more challenging task? For example, the epigenetic modifications. So this technique can tell the difference between all epigenetic modifications in RNA just in the same assay. So I'm using like an uninterrupted data trace to show you the results. And also, this is a piece of tRNA from the yeast cell. So it's a natural compound. Those are natural biomarker molecules containing like a whole lot of epigenetic modifications. And we can do like a epigenetic mapping of this molecule with our technique. And it's very consistent with the true value as well. So eventually, for nanopore proteins, like proteomics, protein is more difficult because it has 20 amino acids and also the PTMs. And these things getting together will get like a really big mess for nanopore. But actually, we all have like a different pore to deal with that. And that is going to have like a more suitable specificity and higher resolution. So this time, we will have like a NTA nickel nanopore. So this nickel nanopore, well, again, using an animation to show you the detail. This is the heteropore, and then we will have the NTA. It will act like a hand, will capture a nickel ion, and this pore will have some metal feature, will capture the amino acid in a highly reversible manner. So this is the event for arginine. So you can see that, remember this feature, you can see that it's very shallow. And then this is the event for glucanate modified asparagine. And it's very different, right? And this is the event for glycine. So technically, all kinds of amino acids will be discriminated by this pore. And again, I'm using like uninterrupted continuous trace to show you the result. So even not even needed for AI, right? So even with your naked eye, you can tell which amino acid it is, right? And also, if we have this kind of high resolution, then why not just go to something even more difficult with PTM? So we have demonstrated like representative PTMs like methylation, acetylation, glycylation, and phosphorylation. Let me just use one example to show you the result. Just with one monosaccharide addition, you can see the difference. And with your naked eye, you can tell you will never make a mistake. Not to mention that we have the help from AI, right? So, and also, like yesterday, we got a paper published by Angavani. So we have further expanded this technique. So all the mirror image amino acids now could be sensed by this assay without a problem. And again, AI is helping by summarizing all these kind of parameters together, and it's now very automatic. And also, because my time is limited, so I will just use my last slide to tell you an interesting story. Besides biology, we can also do like inorganic chemistry. So by engineering this pore a little bit different, and we can use like 2 ligands by sandwiching like a single rare earth element inside, we can actually tell the difference between all the rare earth elements. So even by using this kind of piece of rock, we can generate like a whole bunch of these kind of events. And that's the composition of that piece of rock. So basically, we have developed a technique which has generated like a really rich chemical information as very specialized nanopore and assisted by AI. So we have successfully achieved nanopore analysis of glycans, RNA nucleoside monophosphate, and amino acids and rare earth elements. And even in a very high heterogeneous environment, for example, like a drop of fruit juice, we can tell its composition by using our technique. And very recently, we got a paper accepted, and it's about a drop of water. So even with a drop of water, we can tell the information from it. So that's all for today. Thank you very much.
All right, any questions?
Wonderful talk, wonderful talk. So based on the AI major here, so I'm wondering, is it possible to design an intelligent nanoprotein for your test?
Yeah, that's a very good question. So we are definitely working on that, but actually, we realized that the protein that we already have, which is a natural protein, but we engineered that to be a little bit different from natural, is working really well. And well, from the AI prediction or AI design protein, it's not working as well as this one. So, but I mean, eventually, I think AI will help.
We try to use a large language model to find some like a similar protein, and sometimes we can detect the signal, but we cannot. I mean, we are not good at that part, so cannot.
I think the reason for that is because here this pore has a shape. The shape is not most important. The chemistry is more important. For example, the natural protein will never have a phenylbutyronic acid in its component. So that's why we would prefer to have like a good shape, and then with the chemistry modified, it will become more like immediately usable for us.
Then we have to try different kind of like a modification or something, like you mentioned.
Yes, but actually from the human knowledge, we already have like a lot of chemistry database that we can use to design these pores. And so I think it's chemistry design is easier than biology design, but I think both ways will work.
Thank you.
Thank you.
All right. Very good question. A very good talk here.
Oh, hey, thank you.
So, for my understanding, this kind of model really heavily relies on those standard materials. So you can train your model with clean data.
Right.
So you should very elegant result with all those some fancy new stuff. Do you really have those standard material to train your model, or if I have some new materials need to help me to detect, but I cannot generate those standard material, can you make that model work?
So that's a good point. So first, standard material is not very difficult to access. For example, like a specific kind of amino acids or saccharides, it's all right. So we can train that with pure standard material, and it will generate data that is corresponding to this material. And besides, by doing more and more, and also with AI, we realize that for similar types of materials, they will have something in like similar features as well. For example, saccharides will have their own features. So even without any standard database, by like looking at the data, we will know, okay, sure, this must be like a piece of saccharide. And we can roughly tell like its length or like molecular weight, something like that. So with like more standard data, we will also generate like a model, more advanced model to predict data that doesn't even have a standard database. So I think we already have data to support that, but I'm not showing that today. Yeah.
Thank you. Very good.
Yeah. Thank you.
All right. Let's thank our speaker again.
Thank you.
We move to the last talk of today's session from Defang Ouyang, University of Macau. The title is AI for Drug Discovery, Yesterday, Today, and Tomorrow.
No, it's not good. I think I have just直接放，直接打开看看。
它可能有
取消，也可以取消。关掉。
您自己电脑
我带了。
可以，您自己电脑。PPT 不兼容，我让他拿自己电脑，稍等一下。
Sorry, give us a few minutes. There are some tech issues.
It's okay.
All right, here we go.
Okay, and thanks for the conference organizers. And so what I talk about today is AI for drug delivery. I come from University of Macau, and when we still talk about Macau, everybody think about this casino, right? And however, today we have a very beautiful universities. And what I talk about today, it is first why we need the AI for drug delivery, and then talk about the methodology. And we developed the AI molecular modeling and the PBPK modeling. And finally is case studies. And as we know, the drug discovery can divide into two steps. The first is that we design the new molecules, right? In the past 50 years, the scientists, the developers of lots of computational tools help us to design the new molecules. And however, in the second step, we need to prepare these molecules into proper formulations. And however, we have to do trial and error experiments to do the formulation development. Why is this one? Actually, today formulation development is multi-objective optimizations from high-dimensional space based on the material attributes and the process parameters. We estimate that the formulation space can reach to 10 to 30. That's a very high-dimensional space. However, currently, formulation experiments, we can just do tens or maximum hundreds of experiments. We have to search so high-dimensional space, so that it is laborious, time-consuming, and high cost. And so we think about, can we develop the computational methodology to help us design the drug formulations, just like drug molecule designs? So our group, since 2011, our group developed the data-driven AI with multi-scale modeling, including the quantum mechanics, molecular dynamics, process simulation, and the PBPK modelings together for the drug formulation development. So first, it is the AI drug delivery. And actually, the most difficult in this area, there is no public database for the drug formulations, because all pharmaceutical companies want to hide the formulation as a secret. So we have to build the database by ourselves from the scratch. And also, we developed the rule of 5 for reliable AI drug delivery systems. So we can see this area, before 2018, the annual publication just tens of papers. And however, after we developed the key methodology of AI, and in the last 5 years, this area grows sharply. And last year is over 500. So our group work in this area and have published over 40 the papers. That it is ranked first. Okay, and also combines our past the data and AI model. We built the first the drug formulation AI platform. We call it the formulation AI. That it is contains all marketed drugs and excipients and thousands of data. Currently, this platform has been widely used in the world, including the pharmaceutical company, industry, and academics. This is the first tool. The second tool we worked it is molecular modeling. And as we know, the molecular modeling previously have been used in the CADD with the simple models. However, now we use this molecular modeling in the drug delivery systems with the complex models. And also, our group work in this area since 2011, and also worked lots of different formulations. And however, currently the molecular modeling, the software, it is not designed for the drug formulation simulations. So that is a high technical barriers for the formulation scientists. So recent years, our group worked, it is build a user-friendly molecular modeling platform. We call it the formulation MM, can be used for the formulation very easily. And also recently, we built the physically informed machine learning models. We combine the molecular modeling with the AI model to replace the formulation experiments and get the higher accuracy of the AI models and better generalizations to unseen molecules. So the third tool we worked with is the PBPK modeling to predict the drug in vivo fate. And the principle of this model, it is use mathematical equation to describe each organs of our bodies, and then combine together to form the complex, the models. And however, currently the PBPK models, it is nearly all it is commercialized. The software is high cost, and also is not good for the drug formulation predictions. So in recent years, also our group developed the AI PBPK models to predict the drug in vivo fate. And also, now we work to use this physiology-based AI model to predict the drug exposure and the risk of the inference during lactations, because when during the lactation, the mom sometimes have to take the drugs, and then the drug can diffuse into the milk, that it is high risk to the babies. And the AI model can predict how it works. And the case study we worked with mRNA lipid nanoparticles. So our group built the first AI model for the mRNA lipid nanoparticles in 2021. And also we built the 20 million the ionized lipids libraries and do the visual screening. And we tested 9 molecules and get several positive ionized lipid molecules. That is very interesting. So our group working in this area since 2011. We published first book in this area in 2015, and the second version in the last year. And also, 15 years ago, this area is a very small area with just a few groups working in this area. We organized multiple conferences to encourage more scientists and students to work in these areas. And now AI is widely used in the pharmaceutical industry and also pharmaceutical regulatory agents, including the FDA, EMA, and the CDE. So we can see AI drug delivery from the early stage before 2017, and in recent 8 years, that it is a revolution and a big transformation for the last future. And also we thank our collaborator, our group students, and the funding agency. And thank you.
Time for one quick question.
Thank you, Prof. Can you give us more details about your formulation AI? Yeah.
Okay, yeah, thanks, yeah. And for the formulation AI, that it is currently we contain about 10 different modules, including different formulation modules for the general, different for such as cyclosporine, solid dispersion, and also for the lipsons and other formulations. When you just input the chemical structure of the drug molecules and select the excipients and the amount, and then our AI model can predict how it is the formulation performance, the key critical parameters of this formulation, and then help the formulation scientists can know how it is the formulation performance before you do experiments. Yeah.
All right, let's thank our speaker again and all the speakers for today.
Yeah, thank you.
This is the end of today's session, and the organizer wanted me to inform you guys that there will be a post-award session at the end of tomorrow, so please stay with us. And see you back tomorrow at 8:30? 8:30. Have a good evening.